In this video, we'll be talking about the tetanus toxin, which is produced by the bacterium Clostridium tetani, and how it enters the body and its impacts in our body. This toxin consists of two polypeptide chains, a heavy chain responsible for binding to nerve cell receptors and a light chain that interferes with neurotransmitter release. Let's now look at the modes of entry of this toxin into the body. Another way this toxin can enter the body would be through animal bites. Animal bites from dogs or cats can also lead to contaminated wounds and increase the risk of tetanus if not treated earlier. Traumatic injuries are also another way. A person suffering a cut from a sharp object like a broken piece of glass can develop tetanus if the wound becomes infected. Surgical wounds or post-surgical infections can occur after procedures like orthopedic or abdominal surgeries if the surgical site is not adequately cared for. Grass-on migration is another way the toxin can enter the body. Humans can get grass-ons lodged in their skin while hiking or gardening, which creates an entry point for spores. Some individuals could experience cryptogenic tetanus, which means that they could have developed the disease without a clear history of the wound, possibly due to environmental exposure to the pores. Now you've seen the possible ways this toxin could enter the body. What happens next? Well, let's take a look at where this toxin goes once the body has had exposure to it. After germination of the wound site, the bacteria Clostridium tetani multiplies and produces the tetanospasm toxin in an anaerobic environment, which begins to disrupt normal nerve function. This multiplied growth is often associated with deep puncture wounds where oxygen levels are low, creating a conducive environment for the bacteria. As the bacteria proliferate, they can cause local tissue damage as C. tetani secretes various enzymes such as proteases and phospholipases that can break down cellular components. This damage can lead to increased permeability of the cell membranes in the surrounding area and produce an inflammatory response. Inflammation causes the release of mediators that can alter the permeability of endothelial cells and surrounding tissues. Also, during this time, the bacteria are germinating in the wound, and this is when they release the tetanospasm toxin. Due to the increased damage and increased permeability allows molecules, including the tetanospasm toxin, to leak out of damaged cells into the extracellular space. This gradient can drive the diffusion of the toxin into the surrounding tissue and eventually into the bloodstream. Once in the extracellular fluid, the toxin can be absorbed by nearby capillaries or larger blood vessels. The thin walls of these vessels allow small molecules, including proteins like tetanospasmin, to pass through, and this ultimately facilitates entry into the circulatory system.
After the tetanus toxin enters the bloodstream, it binds to ganglioside receptors present on sensory nerve terminals. By binding to these receptors, the tetanus toxin is able to enter the nerves of the peripheral nervous system. The specificity to enter PNS sensory neurons over other is simply due to their localization near the site of infection. If other neurons were close by, they too could be affected. The tetanus toxin does not spread past the site of infection due to the nitrogen coating around the nerve cells, which attracts the tetanus toxin to them. This flypaper phenomenon was revealed by Kinga Bersensi and her team at UCLA while trying to find a way to deliver new therapies to the nervous system. To gain entry into the neurons, the tetanus toxin interacts with gangliosides on the surface of neurons. These gangliosides are believed to be associated with the tetanus toxin receptor, which, when bound, triggers uptake into the neuron via endocytosis. Once internalized, the tetanus toxin is transported by retrograde axonal transport to the central nervous system. Here, the tetanus toxin is able to reach inhibitory neuron terminals. In a normal neuron, glycine and GABA are released and act on motor neurons to block excitation or release of acetylcholine, which induces muscle relaxation. Let's look at how glycine and GABA act in normal conditions. Glycine and GABA act as inhibitory neurotransmitters in the CNS. GABA and glycine open ligand-gated anion channels with receptors specific to them. Upon activation, GABA and glycine receptors permit the permeation of chlorine through these ligand-gated anion channels. The influx of negatively charged chlorine inhibits postsynaptic cells. The channels that you see in the pictures above are where glycine and GABA bind respectively to trigger the release of chlorine across the membrane into the postsynaptic cell. Therefore, they act as inhibitory neurotransmitters and prevent an action potential from firing and thus relaxation of the muscle. However, when the tetanus toxin is introduced to the bloodstream, GABA and glycine are affected. The tetanus toxin only affects skeletal muscle, while smooth muscle and cardiac muscle remain unaffected by the toxin, which means the toxin only targets the presynaptic motor nerve terminal. Let's pick up where we left off. The tetanus toxin is using the PNS neurons for retrograde transport to the spinal cord. Once in the spinal cord, the tetanus toxin has a choice. Which neurons should it spread to next? Before the toxin can reach its end goal, it encounters excitatory and inhibitory interneurons in the spinal cord. We know its preference for inhibitory interneurons GABA and glycine, but how does it only target these inhibitory interneurons? It doesn't. As the tetanus toxin searches for its next neuronal target in the spinal cord, it targets a specific receptor. Scientist Felix Ye found that this receptor is SV2. He ran a knockout test on SV2 in somatic motor neurons and found that without the presence of the tetanus toxin has no effect. But we run into another problem. As you can see in this picture, both excitatory and inhibitory interneurons in the spinal cord have SV2 receptors, and the tetanus toxin actually affects both excitatory and inhibitory interneurons to some degree. However, the effect on excitatory interneurons is negligible because the tetanus toxin prefers inhibitory interneurons. So how is this preference established? The answer lies in the orthologs of SV2. In a different study, Felix Ye tagged two SV2 orthologs and noted where they concentrated in the spinal cord. He found that SV2A concentrated to GABA and glycine inhibitory interneurons, while SV2B concentrated to excitatory interneurons. Later studies followed this work and tested tetanus toxin's preference for each ortholog. It was found that tetanus toxin has a higher affinity for SV2A and therefore GABA and glycine inhibitory interneurons. This is how the tetanus toxin targets inhibitory interneurons over excitatory interneurons. Once the tetanus toxin targets the SV2A receptor, it binds and enters through a Trojan horse strategy. In normal presynaptic cell, the vesicles release neurotransmitters and intermingle with the membrane before they reform and recycle. During the point of intermingling, the SV2A receptor is exposed to the synaptic junction and the tetanus toxin binds. Then, when the synaptic vesicles are recycled, the tetanus toxin is able to enter the presynaptic cell via endocytosis. Through this retrograde movement from the postsynaptic cell to the presynaptic cell, the tetanus toxin prevents the presynaptic release of GABA and glycine onto motor neurons by cleaving synaptobrevin. The tetanus toxin unbinds from SV2A as it is taken up into the presynaptic cell and attaches to the vesicle membrane where it releases its light chain. This light chain moves through the vesicle membrane and it is able to cleave synaptobrevin using its light chain, which has a zinc peptidase, which cuts a bond in synaptobrevin, leaving it inactive. Synaptobrevin is crucial for the release of GABA and glycine from synaptic vesicles. Without synaptobrevin, the vesicles cannot fuse with the cell membrane and release GABA and glycine, leaving chlorine ions in the extracellular space, resulting in an excitatory postsynaptic cell. When the tetanus toxin blocking the release of inhibitory transmitters, GABA and glycine, the skeletal muscles feel the effect. Since inhibitory signaling no longer occurs, thanks to the tetanus toxin, somatic motor neurons become hyperactive, which can lead to excessive firing. The excessive firing is a result of the excessive release of acetylcholine in the neuromuscular junction. Luckily for the victim, the tetanus toxin does not enter autonomic motor neurons or visceral motor neurons. But why? Through its targeting of GABA and glycine inhibitory interneurons and higher affinity for the SV2A ortholog, the tetanus toxin only affects somatic motor neurons. This is because GABA and glycine interneurons only directly synapse onto somatic motor neurons, which affect muscle cells. If this specificity did not exist, the tetanus toxin would be able to spread to all muscle types and the victim would perish. So the tetanus toxin spreads as it searches for the SV2A receptors, shown here, and binds, preferentially targeting GABA and glycine inhibitory interneurons in the spinal cord, which is why the toxin only affects somatic motor neurons and the victim's skeletal muscles. This is why we see a rigid paralysis induced with spasms of characteristic of tetanus, but no effects of the heart or other organs.
As we mentioned briefly, the lack of inhibitory neurotransmitter release, specifically GABA and glycine, lead to hyperactive motor neurons. But why? Tetanus toxin specifically blocks the release of inhibitory neurotransmitters, but it does not impede the release of excitatory neurotransmitters when the appropriate neurons are stimulated. Excitatory neurotransmitters such as acetylcholine are released into the neuromuscular junction, binding to nicotinic cholinergic receptors and stimulating skeletal muscles. The issue arises from the lack of inhibitory signaling to the skeletal muscles in organisms affected by the tetanus toxin. The lack of inhibitory signals leads to overstimulation of the skeletal muscles and tetany. According to Cleveland Clinic, tetany is a series of involuntary muscle contractions from prolonged overstimulation of motor neurons. The involuntary control and lack of recovery due to suppressed inhibition leads to ventral paralysis of the muscle. As seen in this YouTube video, an individual with a tetany is unable to open their hand and contract it properly due to the paralysis caused by tetany. From analysis done by Penn State, tetanus is typically known as lockjaw due to the characteristic spasms and locking of the jaw one experiences when inflicted with the tetanus toxin, but it has the ability to affect any skeletal muscle in the body due to how it spreads throughout the nervous system. Skeletal muscles include a whole slew of muscles, from muscles in the face, abdominal muscles, muscles in the limbs, and muscles in the neck and back. When inflicted with the tetanus toxin, all of these muscles have the possibility of experiencing overstimulation and paralysis depending on what motor neurons are affected by the toxin and what muscles share neuromuscular junctions with the affected neurons. When the affected neurons share neuromuscular junctions with muscles found in the back, it leads to a condition known as opisthotontis. Opisthotontis, as defined by Stat Pearl's article on opisthotontis, is characterized by a severe backward arched posture due to the overstimulation of skeletal muscles in the back, neck, and lower extremities. Continuous, uninhibited contractions of these muscles lead to their contorted posture. On screen is a painting by Sir Charles Bell illustrating this posture while additionally highlighting other uncontrolled contractions the tetanus toxin promotes by inhibiting GABA and glycine release in the related muscles. Note the contracted arms, hands balled into fists, the clenched jaw, and contracted toes. These episodes of intense, involuntary contractions can cause tears in the muscles, muscle weakness, and fractures due to the sudden contortions and overworking the muscles. While the overstimulation of skeletal muscles that cause opisthotontis have one of the most striking visual effects on the body, overstimulation of other skeletal muscles have further detrimental effects that are not as visible as opisthotontis. The muscles involved in swallowing are skeletal muscles, making the entire process of eating and drinking difficult, as an individual suffering from the tetanus toxin will encounter two major difficulties. First, the overstimulation of the jaw muscles causes the mouth to be stuck, clenched outside the individual's control, as seen in this video here of a man who is unable to open his mouth and quite clearly speak as well as he could. Second, swallowing is a voluntary action, but those afflicted with the tetanus toxin may lose control of the neck muscles involved in swallowing, leading to swallowing difficulties and potentially choking. The inability to control one's swallowing may cause drooling as well. Tetanus toxin does not affect salivary glands, but saliva can quickly accumulate in the mouth if not periodically swallowed. The chest muscles and intercostal muscles are skeletal muscles involved in breathing. While breathing occurs passively, it can be voluntarily controlled, and the contraction and expansion of these skeletal chest muscles allow for the lungs to contract and expand. When the contraction and expansion of these muscles are disrupted, such as when the motor neurons that control these muscles have been affected by the tetanus toxin, the individual may experience difficulties in breathing and may have to rely on respiration machines until the toxin has run its course and degraded.
With the tetanus toxin being taken up into the lower motor neurons and binding to the presynaptic membrane of the neuromuscular junction, the recovery of the synapse is very slow and requires the formation of new nerve terminals and presynaptic structures.
While tetanus toxin have an only half-life of about 5 to 6 days, meaning that it roughly takes 5 to 6 days for the toxin to become half of the concentration from the initial amount, the toxin can be broken in to two different fragments. You have fragment B and C. Fragment C is a non-toxic fragment that possesses an immunological feature that helps neutralize the lethality of the tetanus toxin and is thought to be a key candidate for the vaccine against tetanus. Another way that the synapse recovers is through toxin degradation into two different fragments. You have fragment B and C. Fragment C is a non-toxic fragment that possesses an immunological feature that helps neutralize the lethality of the tetanus toxin and is thought to be a key candidate for the vaccine against tetanus. Another way that the synapse recovers is through toxin degradation. Although there are various ways for toxin degradation to occur, the most likely to occur in this situation would be microbial absorption. This is a surface phenomenon where heavy metals are positioned on the cell surface, and through absorption, the inorganic materials are absorbed into the cell. This mechanism consists of microbes collecting inorganic materials by attaching substrates to them and then absorbing them through the gastrointestinal tract. Once the absorption has occurred, all of the inorganic materials are gone, and it will stop the blocking and release of neurotransmitter at the inhibitory synapse, allowing neurons to once again send signals to other neurons and or tissues. Botulinum, a toxin that also lays in the lower motor neurons and inhibits acetylcholine, is believed that it may reduce the symptoms of tetanus. Botulinum injections can be used in masseter and temporalis muscles to reduce the risk of pulmonary aspiration, anorexia, and dental caries. Just like tetanus toxin, botulinum has a similar mechanism that takes up to nerve terminals of motor neurons, hence activates voluntary muscles. This would be a safe practice for early stages of tetanus due to its more favorable course of the disease, reducing the risk of aspiration and pneumonia. Although there is no information that exists on the use of botulinum toxin on large truncal muscles in tetanus, such as the abdominal or erector spinae muscles, which are seen as general symptoms of tetanus, it helps with the hyperactivity of abdominal muscles and back pain symptoms. The downside to potentially using botulinum as a treatment for tetanus is due to the early stages of the development, with only 6 cases of this being recorded as a successful treatment. Another negative side is how useful it is for most general tetanus symptoms. It is hard for it to treat all muscle groups affected by general tetanus.